Thank you so much, Catherine. It is just lovely to be here. There's such a history of paleontology and paleobotany at the University of Michigan, and so lovely to see so many familiar faces in the audience. So hello, everyone. Thank you so much for having me. So first off, one person does not do science by themselves, and I'm incredibly grateful for a wonderful, wonderful research team, including a lot of phenomenal students. So the Western U.S. work, going back to my PhD time with Peter Wilf and also Scott Wing, continuing to work with them over the years. Reagan Dunn and one of my undergrads, Kimber Circes, helped a lot with this. And then in Ethiopia, as Catherine said, so I started as a postdoc with Bonnie Jacobs and then have continued collaborating with her since then. I'll show some work that a former master's student, Camila Crifo, did, also highlighting Rosemary Bush's work, and is that not the best name for a botanist ever? She got married, and I said to her, Rosemary, you're going to change your name. And she said, are you kidding me? Why would I give up my name? It's too perfect. And then John Wallace, who was an undergrad with me, also working on Ethiopian things. Okay, so I think many people here are very familiar with plant fossils. You have Robin and Selena, but for those of you who haven't visited these two lovely ladies, so here are what some typical plant fossils look like. Leaves, fruits, seeds, also, of course, very important, wood and pollen and spores. And so, traditionally, you find a fossil, and one of the first things that you want to do is try to figure out what it is. Can you put a taxonomic name on it? And sometimes that's quite easy. So this is some work with Aaron Pan and Bonnie Jacobs. We're in a 21.5 million year old site in Ethiopia, found these very distinctive winged seeds, and Aaron, as a taxonomist, looks at these and goes, I know what that is, that's Newtonia. And then he goes to the herbaria, the botanical gardens, figures out, is it actually Newtonia? Is there anything else it could be? And so modern Newtonia, you have this very distinct little groove thing here. You have that little stringer hanging out. And in our fossils, we have that very distinct groove thing, the stringer hanging out. The rest of the morphology works. Nothing else is like that. So we can take this 20 plus million year old fossil and say it is in this modern genus. And then you can go ahead and look at, all right, well, here's where that genus is today versus here's where it was 21 million years ago. And then you can talk about biogeographic changes and what might have caused those. Now, oftentimes, it's very difficult to tell what taxonomically a plant is. We find a lot of dispersed organs. If they're not reproductive organs, it's really, really hard to place them within a phylogeny. But we can still go through those dispersed organs and try to figure out how many species of plant do you have. And so I mostly work in the morphospecies realm. So here's our Bible in the leaf world, describing characters of shape and venation that we can use to distinguish among different morphospecies. So we fill out as many of the characters as we can for a given leaf. And then I've been very lucky to work on sites that have not just the shape and the venation structures preserved, but also that have cell structures preserved. So what you're looking at up here, these are images of the fossil cuticle. So cuticle is the waxy covering on the surface of leaves, very, very resistant to decay. And the cuticle records the imprint of the epidermal cell surface. So three morphospecies that look rather similar at the macroscopic scale, once we have those cellular characters, we're never going to confuse them. So we can look at the arrangement of the stomata. We can look for features like hairs and trichomes. This lovely specimen right here has these papillae in the epidermal cells. So more features to go on is always a good thing. And so once we can divide up all of our leaves into morphospecies, Then we can do things like look at how has plant diversity changed through time, how has the representation of major plant groups changed through time. We can also do things like reconstruct climate. So various techniques that paleobotanists have used. Up here is looking at temperature reconstructions. And in modern forests, there's a relationship between the proportion of species with toothed margins or with smooth margins and the temperature that forest grows in. So a lot of the paleoclimate reconstructions you'll see will be from this. Just for all the real interested people in the audience, this breaks down in the tropics. This is something that works well in higher latitudes, but not in tropical latitudes. Another thing that we'll do is reconstruct precipitation levels, and so that's based on the size of leaves, bigger leaves, wetter climates. And then also reconstructing carbon dioxide levels by looking at the abundance of the stomata on the leaves. So this kind of stuff has been going on in paleobotany for quite a long time. What I want to talk about today are some of the new things that my lab has been doing to try to get at other aspects of the fossil record of plants, and especially at the ecology. And everybody should find Selena later and the awesome things that her lab is doing, also pushing the boundaries of paleobotany and doing really neat, especially the taxonomic and the physiological work. So Selena could give the same introduction, and then she would take us in one direction, and I'm going to take us in a paleoecology direction. So a couple of the things that my lab has been working on are looking at canopy structure through time, at where in the canopy things are growing, and then trying to develop new proxies and apply them to some of the really big questions in paleobotany. So things like the origin of angiosperms. Well, we're not so much doing the origin of angiosperms, but when did they take over ecosystems? And then also looking at forest responses to perturbations and especially to climate change. And then that brings us to what kind of lessons can we get from the fossil record? Can it tell us anything about where we're headed in the next 50, 100 years? So I'm going to start off by talking about some of these new proxies we're trying to develop. And some work a little bit better than others, but that's science for you. Now that I have tenure, I can walk around and give talks about what doesn't work. I might not do that in a job talk, but I can do it now. And then what my bread and butter has been over the years is looking at the fossil record of herbivory on leaves and how that changes through some of these really big perturbations in the geologic record. Okay, so first, if you're going to develop new proxies, you want to go somewhere where the fossil record is awesome, and you don't have to deal with preservation and taphonomy and things like that. So we've been working for about 5 years now in the Mush Valley in north central Ethiopia. So we're up in the highlands. This is almost the extent of the fossil beds right here. So it's this little crater lake, big explosion filled in with water, a very anoxic lake environment, which just preserves fossils beautifully. So here's some of the range of fossils we found with Mulugeta Faseha, who's our geology collaborator at Addis Ababa University. So beautiful, beautiful plant remains, which we'll talk about in great detail. A few insects, and luckily that is enough of a giant termite wing that got Michael Engel really excited, and so he wrote a paper about a new giant termite from Ethiopia. Not very happy fish fossil, and then a very, very happy frog fossil. The frog maybe not as happy, but the fossil is quite happy. So just because they're so pretty. So there's the adult, and we don't just have adults. We have tadpoles. We have tadpoles with legs. We have, I don't know what you call that, middle ontogenetic stage, and then the adults. Matthew Clemens was studying these for his master's degree, working on the taxonomy, thinks that it's a new genus of clawed frogs. And if the hands do have the little claws on them, they're quite cute. And, you know, these fossils are so amazing. So Matthew took them to the University of Texas and used fancy machines there. And we teamed up with Jakob Winther, who's a paleontologist looking at the fossil record of color and interpreting like what color were dinosaurs and birds and all that stuff. So we have the pigments preserved, and Jakob did his magic, and you can figure out what colors these represent, and we had dark gray frogs. So not beautiful tree frogs, but dark gray clawed frogs. Okay, so there's a nice reconstruction of what mush might have looked like with our clawed frog, and there's our Newtonia pod. And we're quite confident that this was a forested environment. So we have, first off, big tree trunks preserved. Then looking at what plant taxa we can put names on, what are the modern descendants of those, or not necessarily descendants, the modern relatives of those things. And the modern relatives are big trees, they're closed forest taxa. We've also reconstructed mean annual precipitation, and that is probably the most important environmental parameter in controlling what sort of vegetation you have. And so there's, you know, modern maps of mean annual precipitation and what sort of plant ecosystem you would get. And our very high precipitation reconstruction puts us in that closed canopy forest. And then Rosemary's work, looking at average carbon isotope discrimination within the mush ecosystem. And there's been modern work done by Aaron Diefendorf and others looking at how this carbon isotope discrimination varies among different biomes, and it does, and our values put us in these closed canopy tropical rainforests. So we said, okay, well, cool, we've got this awesome preservation, it's a closed canopy forest. Can we try to get at where different species are growing in that forest? And can we also get at where an individual leaf is growing in that forest? And so we tried two different proxies. The first one is looking at the carbon isotopes of individual fossils. And so carbon isotopes vary tremendously across different plants. So classic data by Serling from 1997. And I believe it's only grasses that are in here, but if we added other plants, we would see quite a range as well. Now, the first thing that everybody notices is that the photosynthetic pathway is so important in determining your carbon isotope value. So C4 plants are quite a lot heavier than C3 plants. Now, we're working 21.5 million years ago, which is before you have very many C4 plants. So this is probably not very important. We're just looking within the C3 plants. But within those C3 plants, you see a huge range in carbon isotope values. So what is causing that? First off, it could be species. Different species fractionate carbon differently. Plant functional type is also important. So various modern botanists and plant physiologists have looked at like evergreens versus deciduous plants, and angiosperms versus gymnosperms, and you get some significant differences between those groups. And then there's this thing, the canopy effect. So this idea that leaves from different parts of the canopy fractionate carbon differently. Well, not necessarily that they fractionate carbonate differently, but that they have different carbon isotope signatures. And so here's some lovely data by Heather Graham, where she was working in a closed canopy forest in Panama, It's one of those places where they have a canopy crane, so you can go and sample at different levels in the forest. So the top plot is not all that interesting. It's the amount of light versus height above ground. And obviously the higher above ground you are, the more light you tend to have. But looking at this bottom plot right here, so the amount of available light versus the fractionation. And so in lower light regimes, in the bottom of the forest, you tend to have much lighter isotope values than in the highlight regimes at the top of the forest. So isotopes could be telling us, did a leaf grow in the understory or up in the canopy? And so first off, is the isotope signature being preserved in our fossils? Oh, mic is migrating. And yes, I think absolutely. So each point here is an individual fossil leaf. And so Rosemary scraped the cuticle off of that fossil, and then she also scraped matrix underneath where the fossil was. And if we look at, so TOC, total organic carbon, matrix versus fossil, or we could do it by here, separated by species, and we see that the fossils are quite enriched in carbon relative to the rock matrix that they're on. Then looking at the isotope values. So never have we been so happy to see no significant trends. And so it doesn't matter what the matrix value is as far as what the fossil isotope value is. So, signaling that our fossils are giving a distinct signal, and they're telling us what that plant was like, not what the rock around them was like. And so here, separated out by species, you see some differences among species. And one thing that's really nice, so here's our Newtonia, that winged seed, and it has a higher carbon isotope value than all of the leaf types do. And this is something that's observed in modern plants. The fruits and the seeds tend to be a little bit heavier than the leaves are. So a nice little piece of anecdotal evidence to go along with our scatter shots that we think the fossils really are preserving those carbon isotope values from the living plants. Okay, our other proxy has to do with the leaves themselves, the structure, the morphology of the leaves. So these leaves up here come from these same plants, leaves grown in the shade versus leaves grown in the sun. So shade leaves tend to be a lot bigger. They can often be lobier. They're thicker, the sun leaves are thicker. The sun leaves have thicker cuticles. Also, at the micromorphological record, there are differences. And so these experiments were done all the way back in 1942 by a scientist named Watson. And he grew ivy plants in different shade regimes. And we see that the full sun leaves tend to have less wavy cell walls than the ones grown in shade. And this has been replicated for a few other plants, not too many, but and by that I mean this experiment has not been done very many times. So for the few times that it's been done, it seems to work pretty well. Different species have different amounts of differences between these. So this was done back in 1942. We still don't really know exactly why this is. Kind of the best suggestion is that, so you have, your leaf developing and expanding, and then you have the cuticle on that leaf. And as the leaf is developing and expanding, the cuticle is, soft and mushy and able to stretch. And so if something is developing in the shade, then the hardening of the cuticle is a bit slower, a bit patchier, and so that could result in structures like that. whereas in the sun, the cuticle hardens pretty fast, and so the shape of the cell gets set pretty fast and it's pretty homogeneous. So that's the just so story. We don't know if that's true. We don't know about the biochemical pathways that might be leading to that. Okay, so what we thought we would do is we would pick out one species from mush. We picked the most abundant species, which is the legume, and then measure a whole bunch of fossils. And Rosemary measured the isotopes, and my former undergrad, John, measured we call it the undulation index. So he traced around the cells and calculated how sinuous those cells are. Oh boy, okay, so here's our data. So each point is a leaf, so this is the same leaf as that is the same leaf as that, and so on. Undulation index, carbon isotope value, we ordered them based on undulation index. What we had hoped we would see was that carbon isotopes would also have some sort of relationship. We did not see that, and we were really, really sad about this. And this is how science works, and when you get those negative results, it forces you to think harder. Why did we get this negative result? What's going in to setting the undulation index value versus what's going into setting the carbon isotope value? So we sat down and we pondered over that. So factors controlling carbon isotopes, so light affecting how much they're photosynthesizing. Water balance, so affecting when the stomata are open and not open, and is the plant fractionating a lot of carbon. And then water balance is affected by humidity, water availability, branch length, and the height in the plants that you're at. And then also source carbon dioxide value. When is the carbon isotope value set? Well, it's not really set. Throughout the lifetime of a leaf, carbon isotope value can change. And so modern studies have shown a change of 2 to 3 per mil over the course of a leaf's life. Then thinking about what affects undulation index. And this is harder because not as many studies have been done. We don't have a nice study like this for undulation index, like going through a forest and measuring things. So Rosemary's PhD advisor, Francesca McInerney, was really inspired by this data, and she's now in Australia and has students looking at the forests there. And so she's trying to create something like this for those Australian forests and undulation. Okay, so we think light is important. If the story I told you about cuticle hardening being important, then temperature, air current, and humidity would all affect how quickly that cuticle hardens, and thus how much undulation you get. Okay, so some different factors between these two. And then also, when is undulation index set? It's set during the initial expansion of the leaf. So maybe these would have once given us the same signal, and then it changed over the course of the leaf's lifespan. So then, in thinking about these factors, and thinking about the fossil record and what part of the forest we're picking up. So in very few sites are we probably right in the middle of the forest in a true closed canopy. Our fossil record comes from things like lakes and streams, so places where the forest is broken, maybe the edges of forest. And so thinking about, so these are the trees that are dropping leaves into that lake and getting preserved, and how do these factors, well, the ones that aren't in common, differ for a tree at the edge of the forest versus one in the middle of the forest. So we're not quite ready to give up on this yet, and I think there's some really, really interesting things you can do looking at the bulk distribution in the forest, and we found a big range in carbon isotope values, and so we think we are in that closed canopy. and we have a big range of undulation indices, and so we think we are in that closed canopy. But when you try to look at it at the individual level, it's a little messy. So now let's move to something that does work and that we don't have to scratch our heads too much about, and that is looking at the density of veins in a leaf. So, very simply, you take an area of the leaf, you very painstakingly trace all of the veins, get a vein length for that area, divide it by the area, and that's your vein density. And so I had my student Camila did her master's in modern forests and then looking at fossil data. And so the initial goal of Camila's work was to try to tell when neotropical forests originated. When did you get that really closed canopy neotropical forest? And she went and sampled in Panama in closed canopy forests, and then at the Smithsonian Research Station in Maryland, which is a deciduous forest, a little bit more open. And as you'll see, there wasn't a difference between those forests, but we were able to make a nice story out of it in the end. So to get to that story, okay, so angiosperms are very well recognized as having very, very high vein densities. So this is Kevin Boyce's work, and we have our angiosperms down here, these crazy high values that none of the non-angiosperms achieve. And those high values of vein densities were achieved very early in angiosperm evolution. So this is Taylor Field's work, looking at fossils, measuring vein density, and seeing on individual floras how it changes through time. And then we have the non-angiosperms and the angiosperms. So why do angiosperms have such high vein densities? It's correlated with photosynthetic rates and with transpiration rates. So more veins, more transport systems, more photosynthesis occurring. And so that lets angiosperms, really grow quickly, perhaps outcompete some of these other groups and take over the forest. So we were interested in how vein density varied through the forest, and if we could look at distributions of vein densities and see when did angiosperms come to take over the entire landscape. So Camila, she went to these 3 forests, so 2 in Panama, the 1 in Maryland, and she measured individual species, so several leaves per species. In this case and that case, they have canopy cranes, so she could very nicely get lowered into the canopy of the tree and make sure she was getting a sunleaf. And then she could walk around the forest and get the understory. This was trickier. They don't have a canopy crane there. Camila made this lovely system where she had helium balloons and ropes and knives on the rope, and so would use that. So it is an incredible accomplishment that she was able to get 10 species. And this is in a very diverse forest. So we are quite proud of the balloons and knives and getting a sample size of 10 right there. So what you're seeing here, so vein densities versus probability densities. So you can just think about those like histograms. So what's the distribution of vein densities from those canopy species versus the understory species? And we see in all cases that the canopy species have higher vein densities than the understory species. Not surprising, they're intercepting a lot of light, so they're doing more photosynthesis. They're also intercepting a lot of light, so they're in a hot microclimate, and it's good to do more transpiration. We had hoped there would be significant differences among the forests. There are not, so there went the idea of trying to distinguish the neotropical closed forest from a more deciduous temperate forest. But, so there's this black line right there. At this forest, Camilla also sampled the leaf litter. So she took bags of leaf litter, was able to distinguish 19 morphospecies of leaves, measured vein densities in those leaves. And so this is what, what would the fossil record of that forest look like? And first off, a really neat thing is that she captured leaves that have vein densities lower than the lowest canopy leaves. And we like to say in paleobotany worlds that the fossil record is very biased toward canopy leaves. As we saw before, they're thicker and tougher, and there's also more of them. So we thought we're very biased against understory species. But Camilla is getting some of them. And then, of course, she also gets leaves that are higher than the highest understory value, so definitely canopy leaves being picked up, which isn't surprising. So what we decided to do was to take that litter vein density profile from a modern forest, a modern closed canopy tropical forest, and compare it with Taylor Field's data. So the black curve in all of these is the same, and it's that modern vein density profile. And then the histogram represents the species, the fossil species. Okay, so each of these represents a single fossil flora from the different stages in the Cretaceous, and then here's the last stage of the Cretaceous and the beginning of the Paleocene. So big asteroid impact right here. So angiosperms were around before here, so we have angiosperms. Angiosperms are very diverse through all this period, but it's been very controversial about were they important parts of the ecosystem. So we were interested in, so if we compare it to this modern plot, does it seem to say that angiosperms are inhabiting all layers of the forest? Now, very, very small sample sizes, let's not think too much about those, but here we've got 28 species and a very different profile. So perhaps suggesting that those taxa are mostly understory taxa, not big canopy trees. Then we move forward. Gosh, I would love to have more data right here. Our means are very similar, but we don't have the spread, and we don't have those really high values. It's not until the Paleocene when it looks really similar to today. So our hypothesis is that asteroid impact was really important in clearing out the ecosystems. And we talked about how angiosperms have the really high photosynthetic rates, able to grow quickly compared to non-angiosperms. So everything gets leveled, angiosperms get a leg up and take off and dominate everywhere. So we definitely need more Maastrichtian data to test this more. Okay, so we've been talking about the leaves themselves. Now let's talk about the parts that are missing from the leaves. So you walk around your modern forest and you see leaves like these. And then perhaps you catch insects in the act of feeding on the leaves. And we have not just chewing insects, we have insects that make galls. So insect lays an egg in the leaf. Something about that process and hormones or hormones released by the egg cause the plant to make this tumor around the egg and eventually the developing larvae. So the insect very happily lives in here, relatively safe from predators and parasites, goes through its larval phases and hatches out as an advanced larvae or as an adult. Similar idea here. So the female insect laid an egg, that egg hatches, and then the larvae makes a feeding tunnel through the leaf. So it's protected by the leaf, it's eating and pooping its way through, getting bigger, and eventually hatching out. So we see these on our modern hikes through the forest, and we see these in the fossil record as well. So holes chewed in the leaf, you might be asking, how do you know that's a hole and not where you hit it with your hammer? And the answer is that dark rim around it. And so that tells us that the damage was made during the lifetime of the plant. So plants can't regenerate the tissue, but they can build scar tissue around it and make it thick and tough and keep more ripping from occurring, keep nutrients from leaking out. So if we don't see, we call that the reaction rim, no reaction rim, no insect herbivory. And then we have, beautiful galls, leaf vines, etc. Sometimes, very, 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 very rarely, we catch the insects in the act. So we're back at the Mush Valley again, and there's these little scale insects that are preserved on the leaves. And scale insects, so they stick on a leaf. They're often stuck to that one place for their entire lives. They have mouth parts like drinking straws. They puncture the leaf, and they suck out the nutrient goodness. Then sometimes you get very diagnostic feeding patterns. So this is work by Peter Wilf and others, and this is obviously living. It's squishy, it's green. And so ginger leaf, hispine beetle larvae, and there's this very characteristic feeding trail. So it's just scraping the surface of the leaf, and it's avoiding the veins. And we go back 50 million years in Wyoming, and we see that same set of feeding trails, and we can even go back before the giant asteroid impact in the Cretaceous and see that same thing. Oftentimes, though, this is what we're looking at in the fossil record. So beautiful leaves, very clear reaction rims, but gosh, how many insects could do this? Like so many things with biting mouth parts. So we can't tie it to a particular group. But just like we look at species of plants, well, morphospecies of plants, we can look at morphospecies of damage. So holes chewed in the leaf versus things chewing along the margin, galls, mines, different morphologies of holes, different sizes and shapes, different morphologies of margins, and so on. So we can count up the number of different types of damage you see, and that's a proxy for how many different types of insects we're chewing on these. Another thing we can do is with lovely, lovely undergrads, trace the leaves, trace the herbivore marks, and compute what % of that leaf was fed on by an insect. So for the past, I guess almost 15 years now, we've been applying this in Wyoming and looking at climate change and how that affects the plant insect herbivore system. So this is we're looking across the Paleocene-Eocene boundary, a very, very warm time in Earth's history. If you go to southwestern Wyoming, you have the classic, the Green River palms. We could go up to Alaska, northern Alaska, and find these same palms. For the vert paleontologists out there, pick your favorite thing that's restricted to the tropics today, and we find it in Wyoming up in the Arctic Circle. All right, we can in fact be more precise than that. Thanks to the marine record, we have a good quantitative view of how average Earth climate has changed through time. So here we are across the Paleocene-Eocene boundary, and there's this warming trend, and unfortunately, I really need to move that so you can see there's these blue dots going right there. This extreme climate event called the Paleocene-Eocene Thermal Maximum, or PETM. And then the sustained warming in the early Eocene climatic optimum, or ECO. I told you about climate reconstruction using plants, so we've done that in the Bighorn Basin. Here's our climate record. So warming through the late Paleocene, there's the PETM, cooling afterwards, and then warming to the ECO. So I have one of the main PETM players in the audience. I feel like Phil should come up here now, but for those of you who haven't experienced Phil talking about the PETM, so very, very obvious in an ocean core when you hit it. Right there. So we're going forward through time, very abrupt change in lithology, going from limestones to shales, and then transitioning back again. Also very obvious in the isotope record. So oxygen isotopes are telling us about climate. There's an abrupt global warming. And then carbon isotopes have this very abrupt negative shift. So it's a perturbation to the carbon cycle. Lots of light carbon got pumped into the atmosphere, causing warming. Kind of just like we're seeing today, but slower. So very controversial where that carbon came from. Here are some of the hypotheses. I will leave it to other people to argue about those. End result, something like 4,500 petograms of carbon getting pumped into the atmosphere. Carbon dioxide levels rising, something between 2 and 5 times. Ocean acidification, so that's why we're going from those nice carbonate rocks to the clastic shales. Warming of 4 to 8 degrees, depending on where in the world you are. And in the Bighorn Basin, it's about 5 degrees. And then, so that carbon release happens over less than 10,000 years. The entire event is about 200,000 years. Okay, obviously big biotic effects. So the PETM was initially recognized big foram extinction, benthic forams. Then looking at the nanoplanktons, they actually have a big origination event in the PETM. Mammal records, so much of it came out of Michigan, having the first occurrences of many major groups, perissodactyls, artiodactyls, and primates, and things get smaller. So really nice work looking at body size decreasing in the PETM. So Scott Ling and I have been working on the plant record, and then I've also looked at herbivory. So plants, here's kind of a typical Paleocene flora. So lots of conifers and not very diverse. Then we come into the PETM, more diverse, no conifers, and lots of legumes. Then post-PETM, and you look just like you're in the Paleocene again. So here's the data. Each of these is a plant taxa. The PETM is very clearly shown. So all of these things that only occur during the PETM, all of these things that go on vacation during the PETM, not a big extinction event, not a very big origination event. So this is a story of range expansions and contractions with temperature. National Geographic put this together for their PETM article a few years ago. And so we see, pre-PETM, PETM, post-PETM. Plants go back to what they were, but of course the animal trajectory has been forever changed by that event. Okay, so herbivory, we went to some of these best sites of Scott's, including in the PETM, looked at as many angiosperm leaves as we could, looked at what types of damage there were, and looked at how much area was damaged. So first off, looking at types of damage. So here's our temperature curve as a reminder. And let's just focus on the total damage. So very clear correlation between those curves. There it is plotted right there. I will never see R-squared values that high again. And so again, I think this is a range expansion and contraction. And so it gets warmer, more diverse populations from lower latitudes coming northwards, going southwards, coming northwards again. But as I was collecting the PETM sites, it was like, wow, there's a lot of leaves that look like that. I had this gut feeling that there was a lot of herbivory. But I was on a finite PhD timeline. I didn't have time to do all that measuring. And then I became a professor, and I had lovely undergrads who wanted to do that and who got course credit for doing it. And so we decided to do it and to look at whether the area damaged changes during the PETM. So why would it change? Because of changes in temperature and carbon dioxide. So this is kind of mapping out what are the different factors going on. A solid line means it's a direct positive correlation. A dotted line means it's the negative feedback. So plant nutritional quality goes down. Animals would have to eat more to get their nutrition values. So temperature and CO2, have somewhat opposing effects, but it's messy in here. And so I know this, I kind of know that, I'm measuring that, and what's going on in the middle, and can I decouple carbon dioxide and temperature? Well, so first, just to show you the data, so we measured 4 of the sites from the Bighorn Basin, that's these 4, and then we also took data from the literature. These are bulk floras, individual species. And, there is much more damage in the PETM than other time periods. This is looking at museum collections, so they might be biased as far as people either collecting damaged leaves like me or throwing them away like paleobotanists who want to know what the leaves are. So this was an attempt at using those field censuses to figure out what % of the leaves should have damage, and then, we get crazy high in the PETM versus other times. So I told you there's a lot of floral change during the PETM. Is that causing it? Well, we were able to trace one taxa through time. And it occurs in the PETM in the Bighorn Basin. It also occurs in the eco in the Bighorn Basin. I should say that's the eco site right there. And then we also have it at an eco site in the Wind River Basin. And drawing your attention here, the PETM on this same species has way higher area damage than the other time periods. So we spent a lot of time hemming and hawing, and okay, it can't be temperature or CO2 because the eco should be high. Well, what's different between these floras? It's the legumes. We have all these PETM legumes, and we don't have a lot of eco legumes. So I went and looked at, those of you who are wondering, why do I care about legumes? Well, vegetarians know you eat your legumes because you're not eating meat and getting your nitrogen from your meat. So legumes, the good substitute. They have many of them have symbioses with nitrogen-fixing bacteria, and so they tend to have higher nitrogen contents. So that's why we cared. So much more nitrogen-fixing leaves and leaf area in the PETM than in these other time intervals. Well, let's just get everything up there. So we made these beautiful plots of 5 points, and yes, I would like way more than 5 points. I guess there's 6 points on there, but that one's unhappy. And so we seem to see that as you have more of the flora associated with those nitrogen-fixing bacteria, we get more % area damaged. And as we get more of the flora nitrogen-fixing bacteria, the insects don't seem to be preferentially targeting those nitrogen-fixing plants. So if we look at this part of the diagram here, so as CO2 increases, plant nutritional quality goes down. The carbon to nitrogen ratio of the plants increases. More carbon, less nitrogen. So worse food source. But if we have all these nitrogen fixers, they're buffering that. They're buffering the soils. They're providing more nitrogen in that environment. And so that could keep our insect abundances higher and thus our total consumption higher. So would love to have more data. This is the best thing we could come up with. Okay, I am nearly done, but just, exciting things happening. So, PETM we talked a lot about, but there are other early Eocene hyperthermals. So, they were first recognized in the marine realm. Phil, Abel Hemo, and colleagues have been identifying them in the Bighorn Basin. The second hyperthermal, the ETM2, is well known in the Bighorn Basin. I was there this summer, so the lovely age of technology. So I texted Will Clyde, another Michigan product. So text Will coordinate, he says, is it the same level as this? I confirm that it is. He sends me the isotope curve with where I am. And it's a really, really interesting flora. We're nearly done working it up. And we get this mix of normal normal Eocene things. We do have some conifers. We do have some conifers in it. And then we do have legumes in it. We do have very chewed leaves in it. So that is forthcoming. And then I'm colonizing the western U.S., so I live in there. We're spreading out from the Bighorn Basin, trying to work on these other records and seeing, are the things we find in the Bighorn Basin similar to elsewhere? Okay, so where have we gone today? We talked about some new proxies, trying to look at isotopes, trying to look at vein densities, look at cell structure. Some of them works better than others. And then we look at the herbivore damage, we look at these geologic analogs for the present day, and just pointing out, there's hopeful things in what I told you, like we don't have a big plant extinction, but remember that event is so much slower than what we have going on today. And so with that, happy International Women's Day. Please come out tonight to see the Bearded Lady Project movie and talk about science and making it a more diverse and inclusive place. Thank you. Annette, before you leave, it's so nice to see you. So, it's hard. Like the leaf cutter ants, and they make very distinctive, like 180 degree along the side of the leaf. So you see that, you're pretty confident that, it's using it for building a structure, not necessarily for feeding. But outside of that, I don't know how you would. Nathan. We've had this conversation before. No, we have not. Yeah, I mean, one of my dreams, so my first dream is knowing what the carbon dioxide level was. My second dream is knowing what nitrogen concentrations were like in plants. And yeah, I haven't looked at the isotopes. The nitrogen cycle's messy. I've talked with people about, could I do that, and gotten, the nitrogen cycle's messy. I don't know if you could tell like a symbiosis with a nitrogen fixer versus a regular plant. I don't know, do you have any input on that? Yeah. Mm. Right. But well, cuticle's mostly just carbon and hydrogen. Is there nitrogen at all in it? Oh, oh, very interesting. No, no, I have not figured it out. I've been afraid from trying. Afraid of trying. Robin. Right, right. Um, no, I mean, that would be interesting to look at, um, to pump up those sample sizes. And I think, well, I'm not going to try to find it, but Camila did the statistical testing and looking at differences. And I don't remember if she found that a significant difference or not between those two tropical forests. I'll have to look back at that paper. But yeah, I mean, for our African work, we would love to know the, you know, tropical evergreen. Do we use that term? Yeah, versus the more seasonal deciduous. Yeah.
How much compositional difference do you see between the forests you're looking at in the early Eocene, whether it's PETM or later, compared to modern tropical forests, and in relation to the insect damage patterns? In other words, are there differences that
Uh,
might relate to just what species or genera of trees are represented in the ecosystem? I
yeah.
realize it's comparing a lot. It's comparing rich data sets from the past and also from the present. So I'm
Right.
just wondering, or is it a matter that the insects are less? They will just eat. They'll eat. That once they find the right kind of bloom, they'll eat anything.
Yeah,
And so
yeah.
you get very characteristic systemic patterns over a wide range of composition.
Yeah. So Inez and I talked about this quite a bit today. Um, so first off, I don't think we know enough as much about the modern as we think we know, and actually, I'm recruiting.